Hello, I am Chong Lee, the president of EQS and Journal Club manager of QISCA. It is my pleasure to introduce today's Journal Club speaker, Junseo Park from SQRT. Junseo is an undergraduate in science and chemistry at Seoul National University. He is the lab member of a Neuron Goliath Network Research Laboratory led by Professor Sung-Jung Lee in the Department of Physiology and Neuroscience at SNU, and also the lab member of Protein Spin Dynamics Laboratory led by Professor Jung Lee in the Department of Chemistry at SNU. His research interest is molecular mechanisms of neurological disorders in terms of biochemistry. Today, he will be presenting a talk titled Quantum Computing Enhanced Algorithm and Drug Discovery, sharing insights from the paper Quantum Computing Enhanced Algorithm Unveils Potential KRAS Inhibitors by Vakili et al. And BBB-Penetrable Drug Delivery System Modeling by IGF-1R Targeting Chemical Screening by the Junseo Park et al., who is the presenter himself. Please join me in welcoming Junseo to the stage.
Yeah. Thank you for introducing me, and yeah, it's been an honor to presentation in this seminar. And yeah, today I'm going to try to explain about quantum computing enhanced algorithm in drug discovery. And before we started, I introduced myself slightly, and yeah. Oh, yeah. Start now. And introduce myself briefly. And I'm at Junseo Park, and I'm an undergraduate student in Seoul National University. And I define myself as a biochemist. I do in biochemist fields. My major research is like this is first publication, which is submitted in Nature Metabolism, and I want to figure out a molecular mechanism for how like neurological disorder or psychiatric disorder is going to be chronic states. So yes. So my interest in it is, as he introduced me, I want to know the specific kinds of molecular mechanisms. So my base is chemistry, so I use some spectroscopy to analysis like metabolic pathways, some or proteomics like that, and also by validation of some target, then I use in vivo model as mouse experiments by like behavior test, toxic or patch clamp, etc. But in kinds of bridges, I have to use some in silico analysis by some analysis like some which pathway is in instrument things in this some kinds of models, and also I have to make a chemical inhibitor designed by artificial intelligence. So today I'm going to explain about that part in silico analysis and chemical inhibitor design or affinity analysis. So in biochemist, we use artificial intelligence a lot, and I can divide it by two different sections in silico analysis. First is protein structure prediction. It is a Nobel Prize in 2024 in chemistry field. And second is make a new drugs or new chemical for which can make effective like inhibitors or like activator, like we call as agonist or antagonist. So before like design new chemicals or some prediction new chemical, then we have to input some chemical structure into the computer. We know that computer use some vector like number things or some language model like large language model LLM like that. So we have to transform some chemical to which computer can understand. So first is like Smiles, that is very famous tool that use this is Smiles, and there's some kinds of specific grammar to change small molecule compounds to a language model, some sequence. And the second is fingerprint. It is like vector value, and it can find like some hydroxyl as some kinds of vector and some regional structure like that. So we pick some specific a chemical's structure or chemical's property by use a vector value. So most kinds of things use different two fingerprints and smiles. And another thing like molecular graph or imaging is also used in silico drug design. So this first before I introduced traditional in silico designs briefly. This is my research. And next, I'm going to explain that how use the quantum information theory in the drug discovery. So first, it is traditional model. Like this is like I presented in KCS Medical Chemistry Society in last year, and I want to make some chemical like which is IGF-1R is kinds of receptor in brain. I want to make some chemical to activate this receptor. So by synthesis, de novo synthesis is extremely difficult. So I used to in silico screening by like AI-based chemical design. So this is a model plot. I use a smiles as language model and also RDK fingerprint that is also vector. So I combine in the language models and vector to and go to the AI regression models. And IC50 is kinds of affinity value and 50% inhibit some protein. So this is kinds of major affinity value. So and optimizations and make a model which is can prediction IC50 of I make some model, so which input some smiles or some chemical, then the model predicts some affinity by the receptor I pick it, IGF-1R receptor. And second is a design a new chemicals. Like I insert the smiles model data and make a like language model by LLM. And also this descriptor has a lot of fine tuning and make a new chemicals which have a effective high IC50 values. So this is a schematic figures of my model, and I train this as and make a lot of chemicals and also pick. Lastly, I pick the full chemicals which can I predict this can be effective to the IGF-1R activation. But in this case, I picks IGF-1R. This is receptor is extremely famous in my field. So I have a lot of data to insert to computational model. So as I remember, it's more than a trillion like that. Like so, it have extremely large data sets before some kinds of some other researchers made it. So it could be effective because we have a lot of kinds of model data, but most of field, like in this quantum computing research, they use a RAS protein. Most of some chemicals or some protein, which is very effective for pathological aspects, then we didn't have a lot of data. So we have to make a new chemical by the artificial intelligence or some computational things. And also the training by new designed chemical, not real chemical. So it is very hard work. And before that, this work, I use my computer is about I computer is training about a week. It's extremely long day. My computer and goes almost dying, but it's very hard to do a real work at AI-based drug discovery. So, and this I'm not truly sure that, but I know that this is a first paper that used quantum computing by the drug discovery. So the researcher said they use a quantum computing by the making of new chemicals for fine tunings or like making have learning by the artificial intelligence. And this is make a more high affinity or high possibility to make a pro drugs by the in silico model. So they target this is the last protein KRAS is similar by RAS protein. It is if this protein is activation, then cancer goes facilitate. This is we call oncogene, and it only have a 650 data by which is inhibit the KRAS protein. So it is extremely low. We in model of normal traditional artificial intelligence cannot have learning by only 650. So they use a random mutation and more make a more than how can I say that more like some uh one 800,000 molecules by this model processing, and they use, so this is a schematic figures of this model, and yeah, and they pick some 25 million molecule by the visual flow as like kinds of interaction docking affinity, and use a then 25 millions molecule and goes into the QCBM as quantum circuit born machine, and making the molecule and space model and prior molecules by the quantum computational model, and also then making a new chemicals. Then they goes into the traditional artificial intelligence. We call that LSTM model. That is very famous model in AI-based drug discovery, and goes into the making a quantum computing model, a prior model, and goes and input the LSTM model, and they will filter by some chemical 42 model. This is also the open data set, and they they figure out the binding affinity or like toxicity like that in this field. So they make two prodrugs. Like this is they find the 1 and 2. This is prodrug. They find it. Um, so they use a hybrid quantum classical model. This is used like QCBM model, classical QCBM model, like use quantum computing optimizer by like sorry for that. They use hybridized model by they normally we use only making a new prodrug, we use only sequence or vector. So insert the sequence, then computational output a sequence. So but in normal case, sequence didn't truly mean that a chemical is true chemical. So if some sequence, they computer output some sequence, but we have to then we show that it's not truly chemical can realize, or this cannot how to synthesis. So they make a different way to how to make a new data set for the learning and computational. So they have used they use some um make a qubit and 16 qubit, 6 qubit and by IBM hardware, and they made a rotation gates by ratio value and make a rotation on qubits, and also in training gates by C nodes and make a use the data with changing by qubit data, and they use a 3 to 6 layer to depths of this QCBM model. So they insert the small kinds of 650 small data sets to insert this model and make a new similar that data sets like similar chemicals by the language model is here calls that selfies is very similar by Smiles model. So after the QCBM is make a few smart like language like model, then they could be evaluate the chemistry 42 model by then let's see, like this is chemistry 42 model, and model by the protein vegan interaction score, and also the synthesis ability. We can hear more, papers that that's retro synthesis ability, and also some toxicity to use a pharmacological problem. So they filter by descriptor and chemistry 42, and they're changing like fine tunings a few times and make a new prodrugs. And after that, then they really validation that this prodrug is truly activated in vitro or in vivo model. So they is biological experiments also. Like some interesting things is they use NMR techniques. So this prodrug is can effectively binding a RAS protein, and this have like chemical proteins a three-dimensional structure shift. This is all peak as amino acid, and if some binding a chemical, then RAS protein is going to be some peak goes shifted. This is mean that second dimensional structure is changing and this RAS protein is inactivating. So they as validated by the biological things, and this is also affinity or like activate and cell viability test. This is toxicity analysis that, and they suggest that two prodrug and two new chemical by design a quantum computing enhanced algorithm is more effective than the traditional classical language model by AI-based language model. And so yeah, this is I know this is a first paper that truly set the quantum computational things by making a new molecule like molecule descriptor, and this is more effective than traditional artificial intelligence problem. So this is it about data, and so I'm going to wrap it up. So and by the AI-based drug discovery, we have in some biological problem, we can find some proteins or some chemical as effective for pathological problem. So some protein is activate, then goes some neurodegenerative disorder or some cancer is going on. But we have to then we find some protein that we want to make some prodrug to inhibit that pathway. But in here, so nowadays we want to use by not just truly organic synthesis by the laboratory is extremely difficult to. So they want to use by AI-based drug discovery and also some make prodrugs or some potentially activated drugs by artificial intelligence. Oh, wait, yeah, I'm going to start and so so by use a not just traditional language model or like vector prediction, by use a qubit by a ratio and I use a ratio by qubit problem and making it is very this paper show that making a new chemical drugability and drug-like new chemical by the qubit problem is more effective than the traditional AI-based language model. So yeah, this is it. And if you have any questions or someone to talk about it, then please say to me. Yeah, thank you for listening. Um, yeah, and here actually, sorry for this paper didn't explain this is very clearly, but I understand that they just make like how chemical like let's say how like how do I explain it? Wait a second. They said just originally they want to make a input some language sequence and output is also language sequence, but in how to predict or some validations of language to language and make a new some sequence language, then ordinary traditionally they just like large language models, but they combine the QCBM and the middle part, and they make like qubit ratios of the sequence, like how is going is currency 분포. I'm sorry for that. So makes and just the functions up here and also validate by the QCBM analysis. And this is like some kinds of filters on here. So I think in here they use some filters and some generator of new drug-like molecules, data set like data say similar molecules in here. I read some review papers and talking about like other quantum values for drug discovery, but for drug discovery, we didn't kind of normally we didn't kind of like spatial or some time. Just we just want to some chemical is I don't know how, but which is didn't important. Like just effectively binding some protein and protein structure is changing than synthesis. So I think they didn't use some time dependent things or shape spatial things like that. And like and yes. So it's hard to some answer of it. I know this is only paper by using truly quantum algorithm. So I think they have a lot of various way to use combined by quantum algorithm, but I didn't know how other things uses. So there's yeah. Yes. So I guess the point of paper is to demonstrate that you using quantum computer. Yeah, and yeah, they want to say they can use it, and this is more powerful than this ordinary traditional AI model. I think yes, they want to say that. And last protein is not is hard. I mentioned it is cannot be is last protein is we know in biological aspects know that it is very effective target for like anti-cancer drug, but we didn't have a lot of data for in last protein. So I know that the AI-based in silico drug discovery didn't have used this target, but by quantum computing or quantum description, by the more making or more effective chemicals, like then they can do some like a low data problem. They can use it here. So I'm sure you've heard of biotechnology. Yes, yes. Sort of shelf ready. So by your projections, do you think that applying these hybrid classical quantum machine learning techniques, they can possibly potentially offer a much greater speed up than just using currently established deep learning methods? I think like I divided that AI-based in biochemistry, computational chemistry by two areas. First is like you said, protein structure prediction, that is AlphaFold. And I think quantum computation is more effective or have more powerful in like making a new drugs. We didn't have like all answer, just making a lot of various chemicals and can have like affinity binding tests by computational things. But in AI-based protein structure prediction is my own thing, but it is more simple problem. We know the real structure of protein, and we know the sequence of protein. So just combining is, I think, didn't necessary to insert some quantum information or things in like protein prediction area. I think making a new chemical is more hard than just in normal computation. So using some qubits or conjugated a lot of vectors in just simple time, then it could be very powerful. But protein is not that powerful in my things. Is there any questions? I'm going to wrap it up and I'm going to wrap it up here. Thank you for listening my presentation.
Okay, thanks, Junseo, again for the wonderful talk. Okay, let's conclude today's introductory seminar and journal club session. Thank you.